I, Shraddha Shinde from FY Pharmacology, Siagood College of Pharmacy. Today, I would like to present on the topic stress-induced cardiomyopathy, network pharmacology. To start with the introduction, it uses computational power to systematically catalog the molecular interactions of a drug molecule in a living cell. It determines the interaction between the lead drug molecule and specific gene that is target in a disease. Discover new drug targets and leads, and to repurpose existing drug molecules for different therapeutic conditions by allowing an unbiased investigation of the potential drug spaces is the goal of network pharmacology. So steps involved in it are, first of all, the chemical constituents are extracted as leads from Dr. Duke's website. Then it is converted from SDF format to PDBQT format. After that, the first process in the gene extraction is the text mining of genes by PubMed to Ensemble. Further, these genes extracted in this website are added on to gene codes, wherein a geo pathway and KEGG analysis is done. Further, these genes extracted in the KEGG pathway are transferred to protein-protein interaction determination by string database and cytoscape analysis. The cytoscape analysis is further processed, and the genes obtained in it are final, and then they are added on to the molecular docking in Pyrex with the lead molecules, that is ligand. To start with the basic information, the disease is stress cardiomyopathy, whereas stress cardiomyopathy is weakening of the left ventricle as a result of emotional or physical stress. It is also related to myocardial dysfunction, and the excess of stress is developed by catecholamine excess triggered in an acute brain injury. The plant used is Rolfia serpentina. It is an Indian snake root, devil pepper, or serpentine wood is a species of flower in a milkweed family, Apocynaceae. Rolfia alkaloids belong to the general class of medicines called as antihypertensives. They are used to treat the high blood pressure, that is hypertension. Also, it is an effective anxiolytic agent as it attenuates stress-induced behavioral deficits and promotes locomotor activity. So we'll start with the first step, that is chemical constituents of Rolfia serpentine. So we have to search on the Dr. Duke's database on Google and add the plant name in search bar with the selecting the entity type as plant and search it. So these are the chemical constituents that I've got from my plant, with the part that basically selected is root. To run it, so this is the Dr. Duke's website, wherein I've added the Rolfia serpentina. So these are all the chemical constituents that I've got. Excluding the plant, I have only taken the root one, and that all information is gathered over here. So these are all the chemical constituents, 37 chemical constituents that I've got. Further, we have to do open the OpenBabel. It is a tool to convert the file format of chemical constituents 3D structure from SDF to PDBQT format that is required for Pyrex. So, after obtaining these chemical constituents, we have to search it in OpenBabel. So it is fully featured open chemical toolbox designed to speak the many different representations of chemical data. It allows anyone to search, convert, analyze, or store data from molecular modeling, chemistry, solid state materials, biochemistry, or related areas. So first of all, we have to search PubChem. In that, we have to add chemical name and then download the 3D structure in the SDF format. Now we have to open the OpenBabel, and all these single chemical structures that we have downloaded in the 3D format are then converted from SDF format to PDBQT format by the process like. This is the Open Babel website in which uh, here we have to select the chemical compound SDF format, and then we have to say convert, and here we get the output by deselecting the output below only. In this three dots we'll get the format in PDBQT. After the Open Babel, here's the format that we get. So this was the step till the chemical search, and now we'll start with the actual text mining process, in which it is a process by which the genes target involved in the specific disease are extracted from the database called PubMed2 Ensemble. This database extracts all the genes on the basis of information available in the public domain. So steps involved are: open the PubMed Ensemble on Google. Now enter disease name. Select the filter on entries, PubMed IDs. In the database, select Homo sapiens. In attributes, select associated gene name. Select view as all. Click on search. Select all the gene names by excluding the duplicates and add it in the input of gene codes in GeoPathway. So we'll look upon it. So this is the PubMed2 Ensemble database in which I have input my disease name. Here I have selected the filters as associated gene name, and these are the genes obtained. So here if I select all, then all genes name will appear, and these names are then add up to the gene codes GeoPathway. So in this text mining process, a total of 237 genes of stress-induced cardiomyopathy were extracted from text mining process. The next process is Geo and KEGG analysis by gene codes. Geo refers to gene ontology and KEGG refers to Kyoto Encyclopedia of Genes and Genomes. The gene ontology analysis aims to identify those biological processes, cellular locations, and molecular functions that are impacted in the condition studies. KEGG database allows us to understand the utilities and complex functions of a biological system like cells, organisms, and the ecosystem from the level of the genome to molecular information. Steps involved in this process are: search the gene codage database, then select the organism as a Homo sapiens, input type select as gene and proteins. Now select the annotation as GO biological process. Paste the genes obtained from PubMed Ensemble to input column. Now run the launch analysis and obtain the output gene names, gene annotation cluster network, and number of genes involved in the process. So for this, this is the geo pathway I've run here. So these are the genes that are obtained after that. We'll have to search each and every column over here, and then the genes are obtained from each column as in numbers, and these genes are further sorted and they are pasted in the KEGG analysis. Now, after that, in the geo process, I have got 1227 genes of stress-induced cardiomyopathy were extracted from geobiological process in the gene codes. So this is the network that I've got from the geo process, wherein the yellow dots are showing as genes and blue dots are showing as annotations, wherein there are 7 number of genes over there, and in the annotations they are 24 in number. This is the bar chart that is shown over here with the process and type involved of the gene. Same applies for the KEGG analysis. Now change the annotation of gene codes to KEGG analysis. Select all genes obtained in the geo process and add input column of KEGG analysis. Now say launch analysis. Obtain the gene names, gene annotation cluster network, gene description, gene names collected and added into the string database. So as a result of this KEGG analysis, I've got 529 genes of stress-induced cardiomyopathy, which were extracted from the KEGG analysis in gene codes. This is the type of network that I've obtained of the genes and the annotations, wherein 11 number of genes were there and 22 annotations were obtained. This is the bar chart which shows the biological process involved and the pathway of the genes. Moving on to the next process, that is string and cytoscape analysis. The string denotes as search tool for the retrieval of interacting genes proteins, and it is used in protein-protein interaction network determination. String matching module can be used to solve issues or problems regarding genetic sequences and to find the pattern in DNA. Now, the string database website systematically collects and integrates protein-protein interactions, both physical interactions as well as functional associations. Now, the process starts with open string database, select multiple protein option, select organism Homo sapien, add the gene names obtained from the KEGG analysis to input of the string and say search. Output shows the gene characteristic and detailed information on functioning appears. Click the forward tab and PPI network of gene appears. Now download the TSV file format of obtained network and export to cytoscape. Now, this is strange bit database in which the genes obtained from KEGG analysis are inputted in the tab over here. And here we have selected the multiple proteins. Organism is selected as Homo sapien, and the list of genes are added. Here we have to say search. Now, each gene information will appear as in its type, the mechanism of action, and pathway involved. So, all the genes appear over here, and further we have to say as continue. This is the protein-protein interaction network appeared over here, and here we can see that nodes are 48 nodes are obtained. Nodes are the genes, and edges are the interaction of two or multiple genes involved. There are 406 over here. So this strange database structure, we have to convert it into TSV file format. So we can download over here the TSV file format, and then we have to export it into the cytoscape. In the cytoscape analysis, here we have exported from the strange database, and now the genes that are involved are added over here. By selecting the Centiscape menu, we can select the degree and betweenness over here, and the selected genes are obtained. So, as a result of this strange analysis, the protein-protein interaction network of 48 nodes was developed in this strange database. To talk about the Cytoscape, Cytoscape is an open source bioinformatics software platform for visualizing molecular interaction networks and integrating the gene expression profile and other state data. Centiscape computes specific centrality parameters describing the network topology. The centrality parameters aids the users to find the most scientific node in the complex network. The plugin computation generates both numerical and graphical outputs, making easy to find the key nodes also in the large networks. Degree shows the number of connected nodes with the individual node. So higher degree indicates the characteristic of hub. Betweenness centrality quantifies how many times a particular node comes in the shortest chosen pathway between two other nodes. Now, for this, open Cytoscape and TSV file of PPI network imported. Now the degree and betweenness selected from apps. Centiscape 2.2. Network appears with the expected genes involved on the basis of degree and betweenness value. The involved genes are now one by one added to the further PDB. So we'll see about this cytoscape. In the cytoscape, from this tab, we have to select the TSV file format, which I've already selected. In this Scientiscape, after selecting it, in the Scientiscape menu, we have to select the degree and betweenness, and then we have to say over here start. So this is the network that I've got. This is the network, total network of genes. So we can see over here yellow highlighted and blue highlighted genes which are separated, and that is because of the degree and betweenness value. So the according to the degree and betweenness value, according to its limits, the genes that are involved or has the potential to interact with each other are separated. So we'll consider only the yellow highlighted genes over here. Now, in the cytoscape analysis, finally 25 genes were selected based on the degree and betweenness. These are the all 25 genes that were obtained. This is the PPI network that is obtained in the Strange database, and this is the network that is obtained in the cytoscape analysis. Further step is molecular docking. To specifically define it, molecular docking is a key tool in structural molecular biology and computer-assisted drug design. The goal of ligand-protein docking is to predict the predominant binding modes of a ligand with a protein of known three-dimensional structure. It is used to the model interaction between a small molecule and a protein at atomic level, which allow us to characterize the behavior of small molecules in the binding sites of target proteins, as well as to elucidate fundamental biochemical processes. Now, the first step is to download Pyrex, open it, select molecules, add macromolecules, that is the gene 3D structure in PDB format. And next to add the ligand, that is each chemical constituent 3D structure in PDB-QT format. Select all the ligands and one macromolecule at a time and select forward after processing the multiple ligands binding to the gene are shown with the binding affinity score with also multiple binding site. Binding affinity of a molecule to the ligand above -8 or -9 are acceptable. The ligand micromolecule with highest binding affinity could have the potential to show activity for respective disease. And based on the binding affinity, chemistry, kinetics, and dynamic parameters of that ligand could be predicted. So, first of all, the database, the genes is extracted from the cytoscape will have to convert it into PDB format by using the PDB, that is protein data bank. In that, we have to insert or input each protein of each gene obtained, and then 3D structure of that protein is downloaded in the SDF format, in the PDB format. Now, this is the Pyrex. Here, we have to add molecules, wherein we have to select molecules, and these are the ligands which I have already added over here. So, and these are the macromolecules which I have taken. While every run, we have to select all ligands at a time and select only one macromolecule for all the ligands. After that, we have to say forward. So a process will run over here, and after that, we'll get the binding affinity of each chemical compound with the ligand. So this is the binding affinity which I have got here. So as we select it further with forward, and again clicking it forward. This is the binding affinity of every molecule with the genes. So the highest binding affinity is obtained for this first molecule epi-alpha, 3-epi-alpha yohimbine with the gene 1Y4E. So we'll have to compare all the binding affinities, and the highest binding affinity having molecule or the chemical constituent is selected. So after this, we'll have to save this format in the Excel sheet. So by clicking on this tab, we can create an Excel sheet of this binding affinity table. This is the network obtained of the ligand and macromolecule. So according to the table, we can see that minus 278.8 is the highest binding affinity that is obtained. So as a result of the molecular docking, we can say that 3-epi-alpha yohimbine with the 1Y4E gene will have the highest binding affinity and will have the potential of showing activity. The next step is Swiss ADME. Swiss ADME gives the number of parameters and predictive models in order to compute the physicochemical and estimate the pharmacokinetics, drug likeliness, and medicinal chemistry friendliness of one or several small molecules. So, the molecule of interest from the Pyrex is then added to the Swiss ADME. Here, the process involves the uh, from PubChem, we have to extract the canonical smileys of an particular chemical, and then that canonical smiley is added in the Swiss ADME tab. In this Swiss ADME, here is the canonical smiley that we have to paste over here and then say run. So for this molecule, all the database of solubility, physicochemical properties, lipophilicity, drug likeliness parameters are shown over here, wherein we can also see a boiled egg in which uh, it shows the molecule whether it can cross the BBB or whether it will penetrate the membrane or will have the absorption. So the chemical compound obtained has physicochemical properties shown over here with the lipophilicity and solubility, GI absorption, drug likeliness, pharmacokinetics, and water solubility. From all this database, we can conclude that it will have drug likeliness and it will also have water solubility and uh, it will have lipophilicity as in good lipophilic compound. In the boiled egg, we can see that the molecule is present within the BBB labeled structure. That is the indication that it will cross the BBB membrane. To conclude all this, we can say that from molecular docking, it shows that 1Y4E gene with 3-epi-alpha yohimbine has highest binding affinity of minus 278.8. From molecular docking, we could conclude that 3-epi-alpha yohimbine could have potential against 1Y4E gene involved in the stress-induced cardiomyopathy. From Swiss and ADME, we could estimate that the molecule has potential to cross BBB. It is water soluble, it has good GI absorption, and also lipophilic in nature, and has drug likeliness. So this is all about the network pharmacology done of stress-induced cardiomyopathy. These are all the references taken for all the processes involved. Thank you.